Hi, my name is Brenda Perez. I'm a third-year medical student, and today I will be presenting over the opioid-induced changes in the gut microbiota. The opioid epidemic over the last couple of decades has been the deadliest drug crisis in the United States. Americans have the highest rate of opioid prescription consumption in the world. We are aware of the wide range of health complications that develop from opioid use, from severe consequences such as overdose or death, and other less severe but equally as harmful like respiratory illnesses and bloodborne infections. While we know the long-term effect it has on our gastrointestinal system, such as constipation, we are less aware of the impact it has on our gut microbiome. Our gastrointestinal system is comprised of a diversity of microorganisms. Gut microbiome plays a tremendous role in the balance between health and disease. Studies have shown that some of the same mechanisms that are responsible for opioid-induced constipation in humans are similar in rats. This makes it easier to investigate how the ingestion of opioids such as oxycodone alters the intestinal microbiota and the role of microbiome composition on the gut-brain axis. In this study, we used adolescent male and female rats. On day 1, all rats were injected with saline as part of the habituation. On days 2 through 5, saline was injected for the control group and oxycodone for the experimental group. Fecal pellets were collected before and after oxycodone and saline treatment and stored until they were ready for DNA extraction. The fecal DNA was isolated using Zymobiomic mini DNA kit. The V4 hypervariable region of the 16S rRNA gene was amplified using the method of Kozik et al. The 16S rRNA gene-based next-generation sequencing was conducted with an Illumina MiSeq system. Sequences were processed using quantitative insights into microbial ecology. The CLC Genomics Workbench was used for quality control, taxonomic assignments, and diversity analysis. Results indicated that oxycodone treatment leads to modulation of composition and diversity of the gut microbiome. The bacterial composition at the phylum level indicated on Figure A and at the genus level indicated in Figure B are depicted as mean relative abundance. In both samples, Firmicutes and Bacteroidetes are the most abundant. At the genus level, we can see that the abundance of Lactospiraceae and Lactobacillus decreases in the oxycodone-treated group and the uncultured bacterium increases. Figure C shows an example of the longitudinal changes of the gut microbiome composition at the family level in an oxycodone-treated rat. We measure alpha diversity as the observed richness, which is a number of taxa, or evenness, which is the relative abundances of those taxa. We quantify beta diversity as the variability in community composition among samples within a habitat. So in short, alpha diversity measures the diversity within samples, and beta diversity measures the diversity between samples. We measured alpha diversity using Simpson's index. Simpson's index is a measure of diversity which takes into account the number of species present as well as the relative abundance of each species. With this index, 0 represents infinite diversity and 1 represents no diversity. Figure D shows median values of 0.932 and 0.921 in oxycodone and saline-treated groups respectively, which indicates that there is low diversity within each sample. However, the p-value of 0.04 was found with Simpson's index, but no other metrics were found to be significant. As mentioned earlier, beta diversity is used to analyze the temporal and spatial changes in species composition, reflecting whether there is a difference in bacterial communities between groups. In this study, we used the principles coordinate analysis to create a plot of multiple dimensions where each sample is a point and the distance between the points represents the similarity of those samples. So the closer they are together, the more similar they are. Figure E shows a 3D plot measuring beta diversity, which revealed distinct clusters of microbial communities between the saline and oxycodone groups. Permanova was performed to analyze the statistical significance of the beta diversity and showed a p-value of 0.022. Figure F is a heat map analysis which shows few distinct uncultured bacteria induced by oxycodone treatment. You can see the distinct clusters between the two groups. In conclusion, with this study, we were able to explore the impact of oxycodone use on the intestinal microbiome using an animal model. Oxycodone-treated samples demonstrated a shift in the fecal microbiota and its diversity. This is a preliminary study that has laid the foundation for future studies on the consequences of opioid use on intestinal homeostasis, gut microbiome, and the microbiome gut-brain axis. Thank you for your time.